Welcome to Module 2 of this course. This module will talk about the scope and the basic concepts of veterinary pharmacology. For the outline of this presentation, the first part of this module will talk about the definitions of veterinary pharmacology. We'll also discuss the different branches of veterinary pharmacology, the pharmacology-related terms, drug-related terms, and the dose-related terms. This presentation aims to define pharmacology, the concepts of pharmacology, and the pharmacology-related, drug-related, and the dose-related terminologies. It also aims to identify the different branches of pharmacology and discuss their importance. Pharmacology is a term derived from the Greek word pharmakon, meaning drug, and logos, meaning study or science. Therefore, pharmacology simply means a science that deals with drugs. It is also defined as a science that involves all aspects of the action of drugs on living systems. Other references define pharmacology as the study of the therapeutic value and or potential toxicity of chemical agents on biological system. There are different branches of pharmacology. The first branch that we are going to define is comparative pharmacology. Comparative pharmacology is a study of the variation in drug effects in different species of animals. We also have the neuropharmacology. So neuropharmacology is a branch of pharmacology that deals with the study of the actions and effects of drugs on the nervous system. We also are going to discuss cardiovascular pharmacology. So when we say cardiovascular pharmacology, this is a branch of pharmacology that deals on the effects of drugs on the heart, the vascular system, and those parts of the nervous and the endocrine systems that participate in regulating cardiovascular function. The next is the endocrine pharmacology. Endocrine pharmacology is a branch of pharmacology that deals with the study of drugs that are either hormones or hormone derivatives or drugs that may modify the sections of normally secreted hormones. We also have chemotherapy. So when we say chemotherapy, that deals with the use of therapeutic agents in treatment of diseases caused by pathogens. These agents selectively destroy or inhibit pathogens. So the pathogens that we are referring to may include the viruses, bacteria, fungi, and parasites. The next branch is the veterinary clinical pharmacology. This is defined as the clinical evaluation of a new drug for use in animals. It is also defined by Morel and Sams as the study of drug administration and drug effects in animals, many of which are safe or injured. So the science of pharmacology draws on knowledge and methods of many allied clinical and non-clinical disciplines, including chemistry, biochemistry, biology, physiology, pathology, toxicology, and medicine. Veterinary pharmacology is an experimental science dealing with the properties of drugs and their effects on living systems. It has included study of the sources of drugs, which is also known as pharmacognosy, the magnitude and time course of the observed pharmacological effect on the body, which is also known as the pharmacodynamics, the relationship between administered doses, the observed biological fluid or tissue concentrations of the drug, and time in the body, which is also known as pharmacokinetics or PK, used in the treatment of diseases or therapeutics, and poisoning effects or toxicology. Veterinary clinical pharmacology is a subset of pharmacology devoted to the study of the clinical effects of drugs on animal patients with the goal of optimizing the therapeutic dosage regimens. Knowledge of the PK and the PD properties of drugs and their toxic effects is inherent in this veterinary discipline. It is a clinical discipline devoted to the optimal use of drugs in veterinary patients, maximizing their prophylactic or therapeutic benefits while ensuring that the adverse consequences of the drug use are minimized. The demonstration of the efficacy of a drug or a medication includes the following test phases. Number one is the description of the mode of action of the drug. When we say mode of action, MOA or mechanism of action, that refers to the manner in which a therapeutic agent acts, which includes blocking of the receptors, enzymes, stimulating hormone production, and others. Number two is the determination of dose and the dosing intervals. When determining the dosing frequency, the pharmacodynamics of a compound should be considered as critical as the pharmacokinetics. A proper balance should be obtained between dose frequency and size to attain steady-state concentration with minimum fluctuations to ensure therapeutic efficacy and safety. For drugs with wide therapeutic index such as penicillin, larger doses may be administered at longer intervals without any toxic effects. For drugs with narrow therapeutic index such as digoxin, small doses with frequent intervals is better to obtain a profile with least fluctuations. Number three is to conduct dose confirmation trials. And the last is, where applicable, to conduct clinical and field trials. So when we say field trial, this involves the evaluation of whether an agent or procedure reduces the risk of developing disease among those free from that condition at enrollment in the trial. When we say clinical trial, this applies to therapeutic interventions to sick individuals, such as an example of this is the use of chemotherapeutic agents in the trial. For the field trial, that applies to the preventive interventions to healthy individuals. Example of that is the use of vaccine or the vaccine trial. The next are the different pharmacology-related terms. So the first terms related to pharmacology is therapeutics. When we say therapeutics, that is a term describing the treatment of disease in general and includes the use of drugs, surgery, radiation, behavioral modification, and other modalities. When we say pharmacotherapeutics, so pharmacotherapeutics is defined as the study of drugs, drug effects in disease states. It is also defined as the response of an organism to drugs in disease condition. According to the Johns Hopkins University in the year 2012, pharmacotherapeutics is defined as the clinical purpose or indication for giving a drug. Another important term in pharmacology is bioequivalence. So bioequivalence is a clinical term in pharmacokinetics referring to formulations of a drug with rates and extents of absorption that are sufficiently similar that there are not likely to be any clinically important differences with respect to either efficacy or safety. This is applied for formulations with the same active ingredient. Bioequivalence is also a comparison of the bioavailability of two or more products. It is receiving attention because of increased availability and importance of generic drugs in today's pharmaceutical market. Another is the pharmacogenetics. Advancement in molecular biology, especially the recombinant DNA technology, and the emergence of biotechnology has opened fields in pharmacology such as pharmacogenetics and pharmacogenomics. When we say pharmacogenetics, this is defined as the study of the genetic influences on drug responses. It is also the study of the genetically mediated variations in drug responses. For pharmacogenomics, this is defined as the study of the prediction of drug responses between individuals based on their genetic makeup. Let's now proceed to the different drug-related terminologies. When we say drug, that is defined as any chemical agent except food that is used to promote or safeguard the health of animal and human beings. We also have the term over-the-counter drugs. So when we say over-the-counter drugs, these are preparations that can be sold without any prescription because they can be adequately labeled for layman use. We also have the prodrug. So when we say prodrug, those are drugs that are inactive or have a low order of activity in the form administered and are metabolized to the active form in the body. Another important terminology is the hard drugs. So when we say hard drugs, these are addicting drugs capable of producing severe physical or psychological dependence. An example of this is heroin. We also have soft drugs. So soft drugs are less hazardous to health compared to hazard or hard drugs. These are not harmless substances, but the risks are less serious than the risks associated with using hard drugs. So examples of soft drugs are marijuana, sleeping pills, and sedatives. We also have the term placebo. So when we say placebo, this is defined as an inactive substance or other sham form of therapy administered to a patient usually to compare its effects with those of a real drug or treatment, but sometimes to provide psychological benefit to patients by allowing them to believe they are receiving treatments. We also have the dose-related terms. So we'll start with dose. So when we say dose, this is the quantity of the medication to be administered at one time. When we say dosage, this is the determination and regulation of doses. We also have an important term known as loading dose. So a loading dose is defined as a large initial dose of a substance or a series of such doses given to rapidly achieve a therapeutic concentration in the body. We also have the maintenance dose. So when we say maintenance dose, this is defined as a series of relatively small doses that follow the loading dose in order to maintain an effective concentration in the biophase.